Most people think contamination comes from the air. It doesn't. It comes from you. Your hands, your reach, your breath across an open flask. The cabinet is already clean. You are the variable. Cell culture is keeping mammalian cells alive outside a body. You grow them in nutrient-rich media, hold them at 37 degrees in a CO2 incubator, and passage them before they overgrow. Feed, split, repeat.
Simple loop, high margin.
It is not a high-margin loop when you contaminate it.
These cells have no immune defense in vitro. In the body, one bacterium gets eliminated within hours. In your flask, that bacterium doubles every 20 minutes.
12 hours, 1 billion of them, eating your media, poisoning your cells.
It is mildly one-sided.
And the media often looks fine for 2 days before the crash. You won't know until it's too late.
Which is why every single thing you do in cell culture is about keeping microorganisms out, not killing them after. Out. Equipment. Class 2 Biosafety Cabinet. And this is not a fume hood.
A fume hood protects only you. The BSC protects your cells and you. HEPA filtration removes 99.97% of particles 0.3 microns or larger. That covers bacteria and fungi.
The cabinet alone runs 40,000 euros, just for the box.
And worth every cent if you use it correctly. Laminar airflow moves back to front. You work inside that stream. No crossing arms over open flasks. No talking directly into the work zone.
It is mildly counterintuitive that the primary contamination vector in a 40,000 euro sterile cabinet is the person using it.
The airflow is engineered around a cooperative operator. It cannot compensate for someone reaching over an open vessel and disrupting the laminar stream.
Incubator, 37 degrees, 5% CO2, humidified. CO2 is not there to simulate atmosphere. It's buffering your media.
Most culture media uses sodium bicarbonate as a pH buffer. CO2 dissolves into the liquid to maintain the carbonate equilibrium. Let CO2 drop and the media goes alkaline. Cells die.
Humidity stops your media concentrating as it evaporates. Dried out wells kill cells faster than most contaminants. Check the water tray every week.
Wait, a tray of standing water inside a warm incubator? Isn't that just growing fungus?
Yes, which is why it's sterile distilled water, changed weekly, not tap water.
An incubator at 37 degrees is optimal for Aspergillus. The tray can be a fungal reservoir if you're not disciplined. But without humidity, every flask slowly concentrates to toxic levels. Sterile water is the lesser problem.
Sterile distilled water, not expensive, not negotiable.
Starting a session, UV on, 15 minutes before you touch anything, then off.
UV damages DNA, yours included. And once you're working inside, your body blocks the light from reaching the surface anyway. It has no function while you're present.
Fan on 5 minutes before working, flushes any particles that settle during downtime, then wipe everything with 70% ethanol, back to front, never front to back.
Wait, why 70%? Isn't 100% stronger?
70%, not 100%. The water content slows evaporation and allows the ethanol to penetrate the microbial cell wall. Pure ethanol coagulates surface proteins immediately, seals in contaminants rather than killing them.
And spray everything going into the cabinet. Gloves, tube racks, outside of tip boxes. Spray, then wait.
I put the tip box in without spraying it.
It is mildly inevitable.
Take it out. The outside of that box has been on a bench. Spray it 30 seconds, then bring it back in.
The box lid seals the tips internally. They're fine. The external surface carries particulates from the bench to your gloves to everything you subsequently touch inside the cabinet.
Contaminated passage, wasted media, restart, 3 days of work over a tip box.
Before touching a flask, you go to the microscope. Every session. Non-negotiable.
Check turbidity in the media, cell morphology, and confluence. Pink media means alkaline, CO2 is low or cells are starving. Yellow means acidic, too dense, too much lactic acid. Clear to pale yellow is what you want.
If there's contamination, you need to know before you passage it into clean flasks and fresh media. Check first, always.
It is mildly optimistic to assume contamination will announce itself before the damage is done.
Everything touching your cells gets warmed first. Media, PBS, Trypsin, 37 degrees. Water bath, 15 minutes.
Cold media hitting cells at 37 degrees causes a thermal stress response. Membrane fluidity drops, ion transport is impaired. The cells recover, but the recovery introduces biological variability that contaminates your data without leaving a trace.
Tubes in a rack in the bath. Loose tubes tip over, lids come off, bath water gets in.
What's in the bathwater?
You don't want to know.
Accumulated minerals, potential microbial growth, residue from previous experiments, not sterile.
I warmed the trypsin for 40 minutes.
It's degraded. Trypsin is a protease. It eats itself at 37 degrees. 40 minutes in, you've lost substantial enzymatic activity. Your cells won't detach. You'll over-incubate trying to compensate. You'll damage them.
Trypsin undergoes autocatalytic self-digestion at physiological temperature. It cleaves its own peptide bonds at lysine and arginine residues, the same bonds it cleaves in your cell's adhesion proteins. Without serum to stabilize it, that degradation is rapid.
It is mildly self-defeating for the enzyme.
Warm it immediately before use. 15 minutes. Held longer. Discard it and start fresh. Aspirate all old media. Every drop. Tilt the flask. 1 PBS wash. Same volume as your media. Aspirate clean.
You're removing residual serum. Fetal bovine serum contains alpha-2 macroglobulin, a protease inhibitor that complexes with trypsin and neutralizes it before it reaches the cells. Even a thin film of residual media is enough to substantially reduce trypsin activity.
How much does FBS cost?
Around €500 per 500 milliliters for tested mycoplasma-free stock.
And one incomplete wash ruins the whole step.
1 wash is enough. 2 wastes time and stresses the cells. Aspirate clean. Add trypsin. 1 to 2 mils for a T25, 3 to 5 for a T75. Coat the entire surface. Lay the flask flat. Incubator. Check every 2 minutes.
Trypsin cleaves the extracellular domains of integrins, the adhesion proteins anchoring cells to the flask. But it doesn't discriminate. Every minute past detachment, it's cleaving growth factor receptors and membrane signaling proteins. The damage is cumulative and irreversible.
The moment cells start rounding up, add complete media immediately. Full neutralization volume. Serum kills the trypsin on contact. Fast.
It is mildly stressful that the window between detached and damaged is measured in seconds.
Which is why you're checking every 2 minutes and the media is already in the cabinet, warmed, ready to go.
I tapped the flask on the bench to get them off.
Outside the cabinet. Flask open and on a bench. That's a broken sterile zone, a potential crack in the flask, and shear damage on every cell you just tore off the surface.
Mechanical detachment forces the integrin adhesion complex to fail structurally rather than via enzymatic cleavage. The membrane ruptures at the adhesion site. Cells that survive show elevated heat shock, protein expression, and activated stress kinase signaling, a biological artifact baked into every downstream experiment.
It is mildly impressive that one tap introduced 3 separate problems.
If your cells won't detach, the problem is upstream. Residual serum, degraded trypsin, wrong incubation time. Fix that. Don't assault the flask. Transfer to a 15 mil Falcon. Centrifuge 300 times G. 3 minutes. Pellet the cells. Aspirate the supernatant. Trypsin gone. Resuspend in fresh, complete media. Then count.
Hemocytometer with trypan blue. Trypan blue is excluded by intact membranes. Active transport keeps it out of live cells. Dead cells with compromised membranes take it up and stain blue. Count the unstained. That's your viable fraction.
Below 85% viability and something went wrong upstream. Find it before you continue.
What's a T75 flask of HEK 293s worth at that point? Reagents, time, incubator hours.
Factoring in media, Trypsin, FBS, and technician time, comfortably over €100 per passage, per flask.
15% dead cells means you're seeding suboptimal stock. That compounds over passages.
Know your seeding density for your line. It's a number, not an estimate. Too dense and cells hit confluence too fast. Contact inhibited, stressed, behaving abnormally. Too sparse and they lack the paracrine signals to proliferate efficiently. Final thing, mycoplasma.
It is mildly ominous that you saved this for last.
Cell wall-free bacterium, too small for optical microscopy, passes through 0.2 micron filters. Doesn't cloud your media. You will not see it.
If it passes through the filters and you can't see it, how do you even know it's there?
You don't until you test. PCR picks up conserved sequences in the Mycoplasma genome. Doesn't matter which species, the primers catch it.
Studies estimate that 15 to 35% of published cell culture research was conducted on mycoplasma-contaminated lines. It competes with your cells for arginine and glucose, alters gene expression profiles, and affects cytokine secretion. Any assay touching metabolism or signaling is compromised.
It is mildly concerning that a third of the literature may have been written on cells that weren't behaving correctly.
And none of that shows up in the methods section.
Because people didn't test. PCR-based kit every 3 months, straightforward. The alternative is months of data built on a lie.
Mammalian cell culture underpins every recombinant protein therapeutic in clinical use. CHO cells produce the majority of FDA-approved biologics. The technique scales from your T75 to 10,000-liter bioreactors. The principle is identical at every scale.
The global cell culture market is over $25 billion a year. All of it runs on someone, somewhere, doing exactly what we just described, correctly.
And the reason your experiments don't reproduce, the one nobody puts in the methods section, is cell culture. Variable passage number, inconsistent seeding density, undetected contamination. Fix the foundation before everything else. The HEK 293 line I've been running in my shed for the past 2 years is at passage 63. No institutional oversight, no mycoplasma testing schedule anyone else is aware of. 48 T175 flasks. Patar. Viability.
91% counted this morning.
It is mildly concerning that this is not the alarming part of the episode.
Ethanol contact time. Spray, 30 seconds, then handle. Not spray, and immediately grab. The ethanol is still evaporating. It hasn't had time to denature anything. 30 seconds every time. Cell culture. Everything fails because of you, and everything works because of you. Know which one you're being. Method Monday. Every Monday.